amino acids present, but rather it could be many more. But why are we concerned with 20? Because these 20 different types of amino acids are going to be a part of our proteins, right? So the R group in these proteinaceous amino acids could be hydrogen, okay? For example, they are calling it, like one of the examples is going to be glycine, where R group is going to be hydrogen, or it could be a methyl group, as in case of an amino acid called as alanine, or it could be hydroxymethyl group, as in case of serine. So very recently I have told you all, or I had taught you all um, photo respiration, in which I had given you all a shortcut. So I'm sure you all remember the shortcut of photo respiration or C2 pathway. So in that I had given this serine, right? So can anyone tell me that in photo respiration, serine is produced inside which organelle? I just want to know the name of the organelle where glycine is getting converted into serine. Can anyone recollect that? Come on, answer दे दो बेटा. Hmm. Glycine to serine. Which organelle? Photorespiration. Come on, quickly. Yes, yes. Come on. Others, I want all of you all to give this answer. We have completed with photosynthesis very recently. So I'm sure you would be, you know, very good. Yes, right answer. Yes, Jaya, you're right. Yes, yes, Zui. Why a question mark, my dear? Jaya, you're right. Others, come on, others, quickly. सबका गुड मॉर्निंग चाहिए मुझे. हाँ, फटाफट बोलो. ग्लाइसिन टू सेरीन कैसे होता है? आई मीन कहाँ होता है, कैसे होता है छोड़ दो. कौन से ऑर्गेनल में होता है? वाई ओनली टू स्टूडेंट्स आर आंसरिंग? वेर आर अदर्स? Come on, come on, come on. Yeah, you all are right. Why are you putting a question mark? No, Bhumi. No, beta. I told you और फोटो रेस्पिरेशन में 3 ऑर्गेनल्स होते हैं. हाँ, कृष्णा इज राइट. Come on, I want all of you all to answer. Come on, come on, come on. Wake up. 3 organelles. Photo respiration. See, these are the questions which are regularly going to be asked in your examinations. Vidhi, why a question mark? Your answer is right. Niharika, give me one answer. You give me the 3 organelles, but I want one answer. I want one answer. ये 3 में से कहाँ पर हो रहा है ये? Yes, Jaya, you are right, my dear. You are right. The 3 organelles are chloroplast, peroxisome, and mitochondria. So, of course, this particular reaction is taking place. Yeah, right. Now, Bhumi, you are right. So, this particular reaction is taking place in the mitochondria, where glycine के 2 molecules are accumulating in the mitochondria. Once CO2 is lost to the atmosphere, and now the remaining 3 carbon amino acid is going to be called as serine. Okay, so those who haven't answered, please ensure that you revise the content and answer me all the questions which I have, like all the things which have been, already completed with you all. Okay, time flows off. So when that particular R group will be having hydroxy methyl group, then it will be an example of serine. So one amino acid differs from another amino acid not with respect to the carboxyl group or the hydrogen or the amino group. It's going to alter with respect to the R group, that is the variable group. ठीक है, ये important point है. Someone had given me the answer that chloroplast, lysosome and Mitochondria are the three organelles, but a lysosome is having no role in C2 cycle. It's going to be your chloroplast, peroxisome, and this thing, mitochondria. Okay, the chemical and physical properties of amino acid are essentially of the amino, carboxyl, and the R functional groups. Based on the number of amino and carboxyl groups, these can be classified as more acidic. See, basically all are amino acids only. Okay, but basically more acidic. For example, glutamic acid. Okay, basic lysine, neutral valine, but these are all going to be having what? Your COOH group. ठीक है. Similarly, we have got aromatic amino acids. For example, tyrosine and phenylalanine along with tryptophan. These are your aromatic amino acids. ठीक है. A particular property of amino acids is ionizable nature of NH2 and COOH groups. My dears, whatever is being put up on the screen for you all is because a regular group of questions have been asked on these. So which are acidic amino acids, which are basic amino acids, which are neutral amino acids, and which are aromatic amino acids. This is going to be one of the questions for your match the following. If you have seen last year's NEET paper, there are almost 14 to 16 match the following questions. Now what happens in match the following questions? If they ask you just one question, like dash is a acidic amino acid, you will select the answer glutamic acid. So they'll have to give you 4 marks for that answer. Correct? But even if they give you match the following, they'll ask you acidic, basic, neutral, and aromatic. So technically they are going to test your 4 things का knowledge आपको है या नहीं, ये judge किया जाएगा. But you will get only 4 marks for it. So this is a new thing that we have found out in the NEET paper since last year, that they are asking almost 15 match the following. So 15 fours are 60. 60 concepts are being checked just for 15 marks. Otherwise 60 MCQs would have given you 60 marks. But here, because the 4 things are clubbed together as one match the following, you are being tested, you are being assessed for 60 different types of knowledge oriented things. So be very careful. They are doing a lot of smart work, and even we have to do equally the smart work. So which are the, when you read your chapters, this goes as a general rule for all of you all. Whenever you read a particular chapter, while reading, just check it out whether this particular content is a match the following content, or is this particular content can be selected for incorrect correct statement, or can this particular paragraph might be asked for something called as important definitions. So when you read your text, when you read your content, my dears, just don't read it for the sake of reading. Okay, think logically what type of questions could be framed on this content, and once you start making questions in your mind, automatically the answers are going to fit into your brain. Okay, now I guess everyone, like everywhere, morphology of flowering plants chapter chalu ho gaya hai. In that also you can see there are so many examples, and even there, like yesterday only I was talking about aestivation, placentation, phyllotaxy, symmetry of flowers, and all those things. Yesterday only I taught, right? So these are the questions which are sure short question for match the following. So in that case, what are you going to do? You'll not just read, rather it's my suggestion and a request to you all. Okay, wherever you find yes this content could be a question for match the following, immediately आप खुद ही match the following बना लो. with the correct answers. ठीक है, तो जैसे कि राइट नाउ ओनली यू कैन राइट डाउन एसिडिक, बेसिक, न्यूट्रल, एरोमेटिक। एसिडिक के सामने ग्लूटामिक एसिड, बेसिक के सामने लाइसिन, न्यूट्रल के सामने वैलिन, एंड इसके सामने एरोमेटिक के सामने ट्रिप्टोफैन, टाइरोसिन, फिनाइलएलिनिन। So these four things are in front of you as match the following. So while revisions, it will be very easy for all of you all to just quickly look at it and you are done with your match the following content. You need not read the sentences and try to find out what question will be formed on this later on. It will save your time and will help you to revise the content in a very precise way. Is it fine? So my dears, take this type of, if they are the NTA people are smart, we have to be smarter, right? So this is a very good way of remembering the things. Like just now I asked you all that serine कहाँ form होता है, तो ऐसे ये भी मैच द फॉलोइंग के लिए आ सकता है। क्लोरोप्लास्ट, फॉस्फोग्लाइकोलेट, करेक्ट, peroxisome, glycolate, glyoxylate, glycine, right? Chloroplast, mitochondria, glycine, serine. So this is the way you start making, very short. I don't want you to make short notes and all, but at least match the following, select the odd one out, या फिर ऐसे जो 3-4 जगह से ढूंढ के जो क्वेश्चंस बनते हैं ना, start making those precisely as short tricks in your notebooks. It will help you. Genuinely, it will help you. Right? Well, so a particular property of amino acid is the ionizable nature of NH2 and COOH group, that is your amino group, and hydroxyl. Hence, in solutions of different pH, the structure of amino acids change. So here you can see, I think you all know what is Zwitter ionic form. This is what we have said just now. It's this. So here you can see, correct, right? So NH3 इधर पॉजिटिव चार्ज है ना, ये देखो यहाँ पर पॉजिटिव एंड नेगेटिव दोनों चार्जेस आ गए. So this B is going to be called as Zwitter ionic form. जिधर there is a positive charge also and a negative charge also at the two extremes. Did you get me? So this is a diagram which is given in your NCERT textbook. They have written specifically that B is called as the Zwitter ionic form. Kindly do not neglect it. अब इसमें match the following कुछ नहीं है. Here you have to remember the terminology that what is Zwitter ionic form. Okay, so try making your own questions. That is what will help you all. Fine. So that's about your amino acids, because if we have to study proteins, we know proteins are made up of amino acids. Yes, Vaishnavi, you are right. Yes, correct. Okay, perfect. So once we understand what are amino acids, then only we can jump over to proteins, because we know proteins are to be made up of amino acids. Okay? Okay, it's in the form of reactions. It's reactivity. So as we write NADPH2, or we may write NADPH plus H plus, right? These are the different ways that we write for NADPH2. In photosynthesis you might have written about this. NADPH2 also known as NADPH plus H plus. Okay? Proteins, the term protein, that is was given by Berzelius. Now I know this is not going to be asked, so don't worry. This is a content which was initially asked in your AIMS examination. Mulder adopted the term protein to refer the complex organic nitrogenous substances found in the cells of all amino acids of animals and plants. Yeah, this is something important. Proteins are polypeptides. Poly means many, correct? They are linear chains of amino acids linked by peptide bonds. So just now we saw what are amino acids. So one amino acid will be connected with another amino acid to form a peptide bond, or with the help of a peptide bond. ठीक है. How this is going to be formed, right? ये पॉलीपेप्टाइड कैसे बनता है, that is a process which is going to be dealt in detail in the chapter of molecular basis of inheritance. It's a very beautiful process that how this polypeptide is going to be formed. ठीक है, कैसे बनता है वो तब देखेंगे, लेकिन एग्जैक्टली क्या है वो आज हमें समझना है. इफ़ यू अंडरस्टैंड दिस कंटेंट टुडे, in the coming months when we will be going ahead with molbio, it will be very easy for you all to then correlate the things. Fine. So they are linear chains of amino acids linked by peptide bonds. Linear मतलब in one line, एक रो में है, linear है. ठीक है. Each protein is a polymer of amino acid. There are 20 types of amino acids, so they are giving the examples, the names of the 20 types. You will also remember the 20 types के names in molbio chapter. Fine. As of now, alanine, cysteine, proline, tryptophan, lysine, phenylalanine, leucine, there's so many, but total 20, but all 20 you will learn in molbio chapter, molecular basis of inheritance. A homopolymer has only one type of monomer repeating n number of times, just as we have got homopolysaccharides. Glucose subunits were repeated several times. Like that way, if that monomer is repeating again and again, then we call it as homopolymer. But generally it is a heteropolymer. Okay. Dietary proteins are the source of essential amino acids. Dietary proteins means whatever proteins are going to be consumed by us, right? Different sources. So we eat a proteinaceous food, so it's going to suffice the need of what is going to suffice the need of different essential amino acids. So हम सीधा खा लेते हैं उसको, लेकिन कुछ ऐसे अमीनो एसिड्स होते हैं which is I'm coming to it. See, therefore amino acids can be essential or non-essential. So essential are going to be eaten by us. What are non-essential? See, non-essentials are those which our body can make. Technically we require 20 different types of amino acids, but out of these 20, some can be manufactured by our body while some cannot be manufactured. So the one which can be made by our body, though they are important, घर की मुर्गी दाल बराबर, ऐसा है वो, इम्पॉर्टेंट है वो, लेकिन खुद ही बना रहा है ना अपना बॉडी, तो इट इज़ कंसिडर्ड एज नॉन एसेंशियल. प्लीज़ हाँ, दिस इज़ अ वेरी इंपॉर्टेंट पॉइंट. मैनी स्टूडेंट्स थिंक नॉन एसेंशियल मतलब कुछ काम का नहीं है. नहीं बेटा, बहुत काम का है, राइट? But it is manufactured, it is capable of being made in our cells. On the other hand, essentials are those which are going to be taken in as from the dietary sources. ठीक है? So while we get the essential amino acids through our diet or food, that is the reason we should eat balanced diet. ये नहीं अच्छा लगता, वो नहीं अच्छा लगता, ये नहीं चाहिए, वो नहीं चाहिए. हाँ, all these things ना, if you do, you will lack certain essential amino acids. Okay. Well, so proteins carry out many functions in living organisms. Some transport nutrients across the cell membrane, so we call them as transport proteins. Yeah, what we call them as transport proteins means they are helping in transport of something. Okay. Some fight infectious organisms, so what we call them as immunoproteins, yeah, immunoglobulins. They fight infections. Some are hormones, some are enzymes. So different proteins have got different roles to be performed, right? But imagine, for our well-being we require right amount of substances to be passed through the proteins in cell membrane. When we have studied cell unit of life, in that you remember transport protein, tunnel protein, intrinsic proteins, है ना? ये हमने सब किया था. Okay, Yes. Yes, Vaishnavi. So those proteins are coming under your transport. ठीक है, सेल मेम्ब्रेन के थ्रू आते जाते रहते हैं सब्सटेंसेस. So it's so important for our overall well-being. Hormones, proteinaceous hormones. Majority of the enzymes are proteins. I'm using the word majority. Once upon a time, few years before, the statement was all enzymes are proteins, but all proteins are not enzyme. Was few years before. Now it's not like that way. Now I'm using majority of enzymes are proteins. मतलब कुछ ऐसे एंजाइम्स हैं जो नॉन प्रोटीनेशियस है, जिसके बारे में भी हमें आगे देखना है. ओके, वेल, सो कोलाजन, आई होप स्ट्रक्चरल ऑर्गेनाइजेशन आपने सबने रिवाइज किया है, सो यू नो व्हाट इज कोलाजन. Yes, you are right, beta. Carrier protein. Yes, my dear. You're perfectly right. So collagen, I hope you all remember white fibers, yellow fibers. याद है कुछ? So collagen is what? White or yellow? Who can tell me? Collagen is white or yellow? Very good, Arpita. Very good. Yes, Niharika, you are right. चलो चलो आंसर्स दे दो यार. कोलाजन वाइट फाइबर है या येलो फाइबर है? Yes, it's going to be white fibers. Elastin is going to be yellow fibers. Okay, so collagen, the most abundant protein in the animal world, and rubisco is the most abundant protein in the whole of the biosphere. We have seen how rubisco is playing a very important role in C3 pathway, correct? So and C2 as well. Photo respiration में तो rubisco ही तो काम में आ रहा है, right? So I hope you all know what is rubisco. So your collagen in animal world and rubisco in plant world. Okay, they are the most abundant proteins in the whole biosphere. ठीक है, अब ये वाला जो है इन्होंने ये रेडीमेड मैच द फॉलोइंग आपके लिए बना के दे दिया है. कोलाजन फंक्शन इंटरसेलुलर ग्राउंड सब्सटेंस, करेक्ट. ट्रिप्सिन is an enzyme. If you remember digestion chapter, do you recollect something called as trypsin? याद है कुछ? डाइजेशन के चैप्टर में रिएक्शंस किए थे हमने. yes भूमि, राइट. yes Zui, correct. Okay, so trypsin is an enzyme. Insulin is a hormone. I hope you all know insulin for diabetes mellitus. If insulin production is not adequate, the person will suffer from diabetes mellitus, right? So insulin is also going to be a proteinaceous hormone, I would say. Antibodies, IgG, IgA, IgE, right? All these are your antibodies. So antibodies help in fighting infections. So they are the agents which are going to combat infections, I would say. Receptors. So certain proteins act as receptors. So sensory reception for smell, taste, hormones. We have seen already that there are several receptors present in our body. So these proteins even act as receptors. GLUT4, very important. Okay, enables glucose transport inside the cells. So GLUT4 is nothing but it's a protein which is helping the movement of glucose into the cell. So inside the cell means from the bloodstream, the glucose has to transport inside the cell. From the cell cytoplasm, it has to be transported into the mitochondria. I hope you all remember all these things. So once inside the cell, it will undergo glycolysis. Glycolysis will lead to pyruvic acid formation. Pyruvic acid to acetyl CoA. Acetyl CoA will undergo Krebs cycle to ultimately make energy. Right? So the glucose from the blood has to be transported inside the cell cytoplasm. So this is only possible in the presence of this protein called as GLUT4. Is it okay? Have you all understood all these? they are going to, they are not sensitive, they are not, you know, like what are they are going to be what we say, the impulse carrying receptors, the ones who are going to get aggravated or activated, okay, to generate impulse. So that particular receptor on that particular organ is going to be a protein. Okay, got it? So in your ear and all, you have seen there is something called as stereocilias, and the something called as the cochlear part. Okay, no, it does not act as neurotransmitter. The structural part of that uh, sensory receptors is going to be a protein. Okay, got it? So they are the ones who will get activated. Please remember these, match the followings, and the charts. Very important for all of you. Proteins are large molecules containing amino acid units ranging from 100 to 3000, and they are going to have a high molecular weight. In a polypeptide or protein, amino acids are linked by peptide bond, which is formed when the carboxyl group of one amino acid reacts with the amino group of the next amino acid with the elimination of water moiety. This process is called as dehydration. Okay. GLUT4 stands for glucose transport 4. GLU for glucose, T for transport. Okay, GLUT4. Okay. Well, so here what is happening? See, just imagine you have seen that 12 coach rakes, जो ट्रेन आती है अपनी 12 कोच, तो एक कोच के पीछे दूसरा कोच होता है और उन दोनों में एक कनेक्शन होता है. Correct? There are so many people, you know, in rush hours I've seen people travel on that connections also. People sit on that connections also. So in the same way, there's going to be NH2 COH, NH2 COH, NH2 COH. So एक का COH और दूसरे का NH2, इनमें रिएक्शन होकर डिहाइड्रेशन होगा, वाटर मॉलिक्यूल निकाला जाएगा, and they'll get connected. Just as we have seen glycosidic bond formation in carbohydrates, in the same way here also there is removal of water, and this bond between your two amino acids will be called as your peptide linkage, peptide bond. Fine. So the linear sequence of amino acid in a polypeptide chain of proteins forms its primary structure. So what is the primary structure of proteins? A linear chain of amino acids. How many? 100 to 3000. It can vary from 100 to 3000. Okay. Functional proteins have three-dimensional conformation. So this linear protein will undergo modifications. So it will fold and it will refold, and you know, I'm going to talk about it with respect to types of proteins. Water moiety is when OH of one and H of other, okay, OH and H combine together to remove that water. So a potential source where water can be removed is going to be your moiety. Moiety is going to be a part, okay, or a unit. Fine. Okay. So that is going to be your moiety. Elimination of water moiety. Okay. In inorganic chemistry, the structure invariably refers to the molecular formulae like NaCl, MgCl2, etc. Organic chemistry always writes a two-dimensional view of molecules while representing the structure of the molecule, like benzene, naphthalene. Now this is of course your chemistry. Physicists conjure up three-dimensional view of the molecular structures, while biologists describe the protein structure at 4 levels. So every aspect of science has got its own way of expressing the proteins. So in chemistry, like in inorganic chemistry, how do we write? NaCl, MgCl, है ना? We write it in this particular form, na? MgCl2, correct? ऑर्गेनिक केमिस्ट्री में हम कैसे लिखते हैं? बेंजीन, नैफ्थलीन, राइट, कार्बन कंटेनिंग. फिजिक्स के नज़रिया से अगर हम प्रोटीन्स को देखेंगे तो दे विल राइट इट एज molecular structures three dimensionally, and as biologist we explain protein in at four levels. Okay, so we are studying biochemistry with respect to what? Biology. This same chapter is there, biomolecules chapter is there in your chemistry also, correct? But from the biology point of view, we are going to look at the classification of proteins at four levels. ठीक है? The sequence of amino acids, that is the positional information in a protein, which is the first amino acid, which is the second one, and so on, is called the primary structure of a protein. तो एक के पीछे एक जब लाइन लगती है ना amino acids की, तो which is the first one, which is the second one, and so on. So this linear structure of protein will be called as primary structure of protein. Okay? A protein is imagined as a line. The left end represented by the first amino acid, and the last end, the right wala end, is represented by the last amino acid. So 1 to say 120. So the first amino acid and the 120th amino acid. So first amino acid is going to be on the left hand side, while the 120th amino acid is going to be on the right hand side. The first amino acid will have your NH2 group. So N terminal कहते हैं हम लोग उसको. What we call it as N terminal. So the first amino acid is also called as N terminal amino acid. Okay. The last amino acid will be having COOH at the end. So that will be called as your C terminal amino acid. So you know, these are the things which are very basic to understand for classification of proteins. Fine. A protein thread does not exist throughout as an extended rigid rod. The thread, in fact, thread means that linear protein, okay? It will not always sustain as a linear structure. It is folded in the form of helix, similar to a revolving staircase. देखा आपने ऐसा गोल 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 staircase जो होता है वो कैसे दिखता है? Helical. So generally that rod or that thread or that linear protein is going to get helically bound. ऐसे helical बन जाता है. Fine. Of course, only some portions of the protein thread are arranged in the form of a helix. So where there is a turn, that will be a part of the helix. Then again linear, again helical, linear, helical. Okay. Such proteins such as keratin of hair consist of polypeptide chains arranged like a spiral helix. So we all know that hair का protein कौन सा होता है? Hair protein is going to be keratin. So keratin is generally found in a spiral helical fashion. Okay. Such spirals are in some cases right-handed, called as alpha helix, and sometimes it is left-handed, called as beta helix. So if the fold, as I am showing the fold in this form, so मैं right से ऐसे घुमा रही हूँ मेरा जो finger है वो, that is alpha helix. Sometimes it is going to be उल्टा, left sides, beta. Okay. The spiral configuration is held together with the help of hydrogen bonds. Now, who holds this spiral or this coiled structure? Someone has to be there to keep the spirals or keep the helices at place. So, उनको पकड़ के कौन रखता है helical form में? Hydrogen bonds. Fine. In proteins, only right-handed helices have been observed. So, it could be left-handed amino acid orientation, but proteins are generally going to have right-handed. Did you all understand this? Look at the beauty of this. Edge bonds will be present at every turn. Edge bond will be, like for example, you can see this wire. Okay, this is the wire. It's a linear one, correct? If I have to turn it like this way, हाँ, तो इसको और इसको जब तक कोई पकड़ेगा नहीं तब तक वो हेलिकल नहीं बनेगा ना. इट कैनॉट बिकम स्पाइरल, राइट? सो समवन हैज टू होल्ड इट in place, otherwise again it will come like this way. So wherever there is going to be a turn na, wherever there is going to be a helical structure formed na, there will be hydrogen bonds which will help the helical structure to remain in this particular helical position. Understood where the hydrogen is going to be present? Okay, yes, fine. Well, then in some proteins, two or more peptide chains are linked together by intermolecular hydrogen bonds. So now imagine one thread which is red in color, one thread which is blue in color. And when red and blue threads will be held together using what? Intermolecular hydrogen. It's not intra. Intra means within the same chain. Inter means within the two different chains. Okay, so that is, yes, 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 yes. We are seeing the structures. Yes, yes, yes. That is going to divide the proteins into primary structure, secondary structure, tertiary structure, and quaternary structure. Okay. So in some proteins, two or more peptide chains are linked together by intermolecular hydrogen bonds. ठीक है? Intra, within, inter, between. ठीक है? Yeah. So such structures are called as beta plated sheet. बीटा प्लेटेड शीट कहते हैं उनको. आगे मैंने डायग्राम दिया है, फोटोग्राफ दिया है जिसमें ये बहुत क्लियरली दिखाई देगा व्हाट इज बीटा प्लेटेड शीट. So beta pleated sheets are the structures which are found in proteins of silk fibers. So my dear, see here, you know, to make the thing simple, examples भी दिए हुए हैं. So keratin was helical protein. Beta pleated is found in silk fibers. We wear silk clothes ना, silk कुर्ता होता है, silk sarees होती है ना, silk के dresses भी अच्छे खासे मिल जाते हैं. So that silk fiber is going to be having what? Beta pleated sheet. So that makes us very clear that silk is going to be a protein, right? In large proteins such as myoglobin, where is myoglobin? बताओ, मायोग्लोबिन कहाँ का प्रोटीन है हमारे बॉडी में? यस, वेरी गुड जोई, कम ऑन अदर्स. हाँ, ऑल हैव आंसर्ड. वेरी गुड. बताओ बताओ, बाकी के लोग बताओ. यू जिन्होंने आंसर किया अब तक सारे सही आंसर्स हैं. मायोग्लोबिन कहाँ का है? Heme containing protein is hemoglobin. I am talking about myoglobin. Come on, we have done the chapter of structural organization. Yes. Of course, yes, yes. Palak right. Yes, Krishna was also right. Yeah, it is about the muscle. Okay, it's the muscle fibers. मसल का प्रोटीन है वो मायोग्लोबिन. So in large proteins such as myoglobin and enzymes, peptide chains are much looped. Looped means they are going to be just making, you know, several loops बनाते हैं, guard बनाते हैं. Okay, they are twisted, they are folded back on themselves due to formation of disulfide bonds. It appears like a hollow woolen ball. So many a times what happens is these large proteins na, they have got these linear polypeptides wound on themselves like a hollow woolen ball. आपने woolen balls देखे हैं? Woolen ball क्या होता है पता है? What is woolen ball? हाँ, तो ये ना ऐसे ही दिखते हैं, woolen balls के जैसे दिखते हैं. So who keeps them in that woolen ball like thing? The disulfide bond. So along with hydrogen bonds, there's also going to be your SS bonds, disulfide bonds. Okay, such loops and bends give the protein a tertiary structure. So primary structure was a linear one, secondary structure was spirally or helically coiled along with beta pleated sheets, that is your secondary, and tertiary structure of protein is like a hollow woolen ball. ठीक है, तो प्राइमरी स्ट्रक्चर में सिर्फ पेप्टाइड लिंकेजेस थे, सेकेंडरी स्ट्रक्चर में हाइड्रोजन बॉन्ड्स भी थे, और टर्शरी स्ट्रक्चर में पेप्टाइड बॉन्ड्स है, हाइड्रोजन बॉन्ड्स है, एंड अलोंग विद दैट डाइसल्फाइड ब्रिजेस भी है. अंडरस्टूड? ओके. Tertiary structure is absolutely necessary for many biological activities of proteins. So majority of the proteins are going to be functional only at tertiary level when they become tertiary proteins. Is it fine? Have you all understood primary, secondary, tertiary till now? There is one more, that is quaternary structure. But before I go to the fourth one, these three, all of you all are okay? Primary is going to be linear, secondary is going to be spiral or helical, of course even pleated is going to be secondary only beta, tertiary is going to be like a hollow woolen ball. Okay, in primary only peptide linkage, in secondary peptide linkage along with hydrogen bonds, in tertiary peptide linkages, hydrogen bonds and disulfide linkages. These are the key points. ठीक है. Some proteins are an assembly of more than one polypeptide or subunits. Okay, so one polypeptide, along with that one more polypeptide, or more than, like two also. So the manner in which these individual folded polypeptides or subunits are arranged with respect to each other. For example, they could be linear strings of sphere, or they could be spheres arranged one upon each other, or they could be in the form of a cube, or they could be in the form of a plate. Like different types of arrangements have been found. It is the architecture of the protein otherwise which will be called as quaternary structure of protein. For example, hemoglobin. So adult human hemoglobin is going to be having 4 units. 2 of these are identical to each other, hence 2 subunits of a type, and of A type you can call, and 2 subunits of B type together constitutes the human hemoglobin. So hemoglobin is going to be a quaternary structure. Okay, so 1, 2, 3 and 4. So there are 4 polypeptides which are arranged together to form one unit. Okay, so primary, secondary, tertiary and quaternary, we have got 4 levels of classification of proteins. ठीक है, look at this. Yes, it will make the things very simple. So this is your primary. देखो एक के पीछे एक डिब्बा लगा है. Correct, this is your primary, which is a polypeptide. This is secondary. Here you can see helical structures. Now someone was asking where exactly is the hydrogen bonds. So hydrogen bonds will be present between this particular area to keep the loops intact. Okay, यहाँ पर, फिर यहाँ पर, इन दोनों के बीच में. तो जहाँ जहाँ फोल्ड्स है ना, जिनको ऐसे पकड़ के रखना चाहिए, वहाँ पर है. So this is called as your alpha helix. This is your beta pleated sheet. ये देखो. Yes, they act like clips. Yes, very good. Yes, Vaishnavi. These are your beta pleated sheets. Just look at it, how they are. Okay. Tertiary देखो, hydrogen bonds भी है and disulfide bonds भी है. So it appears like a woolen ball, hollow woolen ball. This is your tertiary. And look at this quaternary structures. So this is one chain of polypeptide. This is another, which is almost intermingling with each other. This is your A type of structure. This is your B type of structure, and they are held together as a quaternary structure. Okay, so primary, secondary के 2 types है. Either it will be helical structure or it will be pleated structure. Tertiary, hollow woolen ball, and quaternary, more than one polypeptide chains associated with each other in different forms. Okay, disulfide bonds will be present over here. See, between the 2 folds. to overlap. जैसे कि for example you take a kerchief, handkerchief. हाँ, कर्चिफ लेते हो तो कर्चिफ को अगर आप एक फोल्ड करोगे, if you fold your handkerchief into one half, that will be your secondary structure. You make it into one more half, तो रेक्टेंगल का अब स्क्वायर बन गया. ओके, तो दो हाफ्स को ज्वाइन करने वाला हाइड्रोजन बॉन्ड है, और वो रेक्टेंगुलर शीट को अब स्क्वायर में बनाने के लिए जो बॉन्ड्स आएंगे ना, दैट विल बी योर डाइसल्फाइड. अंडरस्टूड? आई गेस आई विल बी हैविंग कर्चिफ समवेयर. कर्चिफ है, हैंडकर्चिफ है कहीं पर. Yes. So see, it's very simple. So as you can see, this is one kerchief, right? So this is your linear. When I fold it, see, this is a rectangular. So इसको rectangular बनाने के लिए मैंने ये जो मेरे दो हाथ यूज़ किए हैं, दो points जो यूज़ किए हैं, that is of course going to be my hydrogen. And when I again make it into a square, right? So यहाँ पर जो bonds है ना, they'll be your sulfides. Okay, like this way there will be, if I take a red, like a blue color ka kerchief, okay, then that two together, if I join them together, blue with white, then it will be quaternary. Yes. Even quaternary will also have three types of bonds. Agreed? Yes. So no confusion now. Yeah, this is a simple version of drawing it. So a secondary structure loops this alpha helix, and this is tertiary structure, like a hollow woolen ball. Any difficulty in understanding these points? Anyone who has not followed these points? Easy? I'll go ahead. Proteins are extremely reactive and highly specific in behavior. Proteins are amphoteric in nature, that is, they act as both acids also and bases also. So please remember this scientific term called as amphoteric. So what exactly do you mean by this word amphoteric? That particular component which can act as an acid also and which can act as a base also. ठीक है? The behavior of protein is strongly influenced by pH. Like amino acids, proteins are dipolar ions at the isoelectric point. What do you mean by this? That is, the sum of positive charges is equal to the sum of negative charges, and the net charge therefore will be 0. ठीक है? इसको हम क्या कहते हैं? Dipolar ions at isoelectric points. So जितने positive charges हैं उतने negative charges होंगे. Net charge on proteins would be 0. Okay. The ionic group of proteins are contributed by the side chains of polyfunctional amino acids. So there are many amino acids which is going to have side chains with positive or negative, charges on them. So when we talk about molecular basis of inheritance, we'll be coming about histone proteins, and in histone proteins we know that they'll be having positive charge on their side chains, okay, and therefore they are going to attract the negatively charged DNA. Don't worry, little bit we have done in cell unit of life, much more we have to do in molecular basis of inheritance chapter. But till here have everyone of you all understood very precisely? Any difficulty with proteins? Nothing? A protein consists of, that is what, isoelectric point is the one which is having net zero charge. जितने पॉजिटिव चार्जेस है उतने ही नेगेटिव चार्जेस रहेंगे, बैलेंस आउट हो जाएगा, एंड नेट जीरो चार्ज, दैट इज आइसो इलेक्ट्रिक. आइसो मीन सेम. So when both the charges of positive and negative become same, it's the isoelectric point. Okay. Yes, NH3 plus and COH minus. Correct, my dear. Perfect. Very correct. Okay. A protein consists of more basic amino acids such as lysine and arginine, which exist as a cation at the physiological pH of 7.4. Such proteins are called as basic proteins. ठीक है. Histones of nucleoprotein are basic proteins. So just now I was talking about histones only, correct? A protein rich in acidic amino acid exists as an anion at a pH which is going to be, of course, acidic. So that pH is going to be your around 5.8. Okay, physiological pH would be in which they will be activated. Such proteins are called as acidic proteins. Most of the blood proteins are acidic proteins. Now see, histone proteins of nucleic acids are basic, while most of the proteins of the blood are acidic. Again, very important points. ठीक है. If we start classifying the proteins on the basis of structure, proteins are classified into three categories. Simple proteins first. They are simple proteins which are going to be formed by hydrolysis, which will yield only amino acids. Okay, so when we hydrolyze these simple proteins, we'll get only amino acid moieties. We'll not get any other type of any other type of chemical present over there. It's only amino acids. ठीक है. They are soluble in one or more solvents. Simple proteins may be soluble in water as well. Histones of nucleoproteins are soluble in water. That is your histones are part of your nucleic acids, DNA. Globulin molecules of histones are not coagulated by heat. Albumins are soluble in water, but they get coagulated on heating. You know, albumin is going to be your egg white. Egg white जो होता है, it is going to be loaded with albumin proteins. So when you fry the egg, okay, when you just break open the egg and put it on the वो तवे पे डाल देते हैं, तो वो coagulate हो जाता है. What is coagulation? You know ना? It's going to be completely wrinkling out. So they get coagulated on heating. Albumins are widely distributed. Best example is egg albumin, serum albumin, legumulin of pulses. They all are albumins. So those who are eating egg omelets and even boiled egg, that white part of the boiled egg is actually a coagulated albumin protein. So many a times it's said that you should not eat that yellow part of the egg, but you should always eat the white part of the egg. It is rich in proteins is what we are generally told. Come on, जो लोग जिम के लिए जाते हैं, बॉडी बिल्डर्स, anyone over here who are into bodybuilding? Huh? Yes, obviously. Those who want to have a very good muscular strength and a very good, body building के लिए जो actually generally like जो जाते हैं, they are regularly consuming egg whites. They don't consume the yellow part. Yeah, exactly. Yellow is loaded with fats. Yes, so yellow should not be consumed, but white should be. Yes, wait, you're right, beta. Perfect. So yeah, Krishna, even you are right. Yes, so that is what I want to tell you all, that like these are all sources of proteins. So when you eat legumes, what are legumes? You know ना, मॉर्फोलॉजी में हमने अभी नहीं किया है अभी तक. कुछ legumes के बारे में हमने किया है example. Yes. Anyways, so your pulses, your legumes, they also are very good source of proteins. So in legumes, soybean is the richest source of protein. So those who are vegetarians and not even vegetarians, for them generally pulses are very good source of proteins. चना, छोले, हाँ, फिर अपना सोयाबीन, राजमा, loaded with proteins. Fine. Okay. Second type is going to be your conjugated protein. So first classification was what? Simple protein. Second one is going to be conjugated protein. So what do you mean by the word conjugated? Conjugated means two things coming together. Yes, yes, Shwet. Yes, you are right. Even drumsticks is loaded with proteins. I agree. Obviously. Conjugated protein consists of simple protein in combination with a non-proteinaceous substance. So protein plus non-protein is conjugated protein. For example, hemoglobin. See, the non-protein group, which we call it as prosthetic group, is heme, right? And globin is your protein. So globin is the protein and the iron containing pigment heme is the prosthetic group. Okay, so what is a prosthetic group? A non-proteinaceous substance which is attached with simple protein. That is your prosthetic group. Okay. Similarly, nucleoproteins have nucleic acids coupled with histone proteins. So histone क्या है? प्रोटीन है. न्यूक्लिक एसिड क्या है? डीएनए है, आरएनए है. हाँ. So when they two will be combined together, we call them as conjugated proteins. Okay. On the basis of on this basis, proteins are classified as even glycoprotein, mucoprotein. Yes, yes, yes. Those entangled ones which you saw just now, were your hemoglobin molecule. That was your hemoglobin proteins. Correct. Mucoproteins are carbohydrate plus protein. Okay. Mucin of saliva, heparin of blood. These all are your mucoproteins. Fine. लिपोप्रोटीन्स सुने ये सारे नाम ना. लिपोप्रोटीन नाम से पता चल रहा है लिपिड प्लस प्रोटीन. So lipid protein complexes conjugate proteins which are found in brain, plasma membrane, milk, all are rich sources of lipoproteins. Fine. So first was simple proteins, second is conjugated proteins, and third are derived proteins. Now what do you mean by derived? See, these proteins are not found in nature as such. So original natural form में ये proteins नहीं मिलते हैं. Rather, these proteins are derived from native protein molecules by hydrolysis. So peptone, metaprotein, these are derived proteins. You remember peptone in digestion chapter? याद है? कुछ याद है ये word peptone? Exactly. So it was what? It was a product of hydrolysis of protein. Okay, we call them as partially digested. So these are your derived proteins. They are not going to be found in nature. They are derived. So three types of classifications: simple, only amino acid; conjugated, protein plus non-proteinaceous substance; and derived, not found in nature. They are derived after hydrolysis. Best example are metaproteins and peptones. Peptones तो याद रहेगा. कोई डिफिकल्टी है बेटा क्लासिफिकेशन में? Anyone having any problem in understanding proteins till now, whatever we have done? They are also derived proteins. They are also derived. Okay, they are not found in nature. They are derived. Okay, they are generally added for in wheys and all. जो whey powder खाते हैं ना लोग, again for bodybuilding, या फिर protein shakes पीते हैं, they add. It's a supplement. Fine. That's all with the proteins. Okay. Now we come to nucleic acids. Nucleic acids is going to be your, my dears, a request to all of you all. It's a humble request to all of you all. Kindly pay very good attention to this part of nucleic acids, because the same thing will be again repeated in my molecular basis of inheritance, and that time no one is going to teach you the basics of nucleic acid, because it is assumed that ये तुम लोग को अभी अभी पढ़ाया है तो आपको ये आना चाहिए. ओके, प्लीज़. तो ये पार्ट को बहुत अच्छी तरह से समझ लेना. नहीं समझता है तो पूछ लेना बार बार. But I want all of you all to pay your 100% attention to this nucleic acid part. It's very crucial. Okay. No beta. Metaproteins are generally not naturally formed also. Okay. they are going to be made to get that particular metaproteins. Fine. Nucleic acids, Swiss biochemist Friedrich Mischer, remember his name, it has been asked, discovered and isolated nucleic acid from the pus cells of an owl. I guess we had discussed this in Cell Unit of Life. By 1938, became evident that nucleic acids are of two types, deoxyribonucleic acid and ribonucleic acid, that is DNA and RNA. DNA is found in chloroplast and mitochondria, along with your nucleus, of course. DNA is, of course, the hereditary material in most of the organisms. When they say most of the organisms, exceptions are viruses. So in several viruses, RNA is going to be the genetic material. The nucleic acids are among the largest of all molecules found in living beings. They contain three types of molecules. A 5-carbon sugar, जिसको हम पेंटोज शुगर कहते हैं, phosphoric acid H3PO4, nitrogen containing bases, that is adenine, guanine, cytosine, uracil and thymine. Three join together to form nucleotide of nucleic acid. I guess these things should be known to you all, because what is ATP करके मैंने हर एक क्लास में आपको पढ़ाया था. मतलब in all batches I had taught what is ATP, and that time I had drawn this 5-carbon sugar. I had also drawn the phosphate molecule कहाँ होता है, and I had also drawn the base, nucleotide base कहाँ पर होता है. So I guess you should know it, but still. Living organisms have a number of carbon compounds in which heterocyclic rings are going to be formed. Some of these are nitrogen bases. They are heterocyclic rings. Okay. Example: adenine, guanine, cytosine, uracil and thymine. So what are these? These are nitrogen containing compounds. Okay. जिसको हम nitrogen bases कहते हैं. Okay. When found attached to a sugar, we call them as nucleosides. So a pentose sugar along with a nitrogen base, these two will together be called as nucleosides. Okay. If a phosphate group will be attached to the nucleosides, then we call these three things together as nucleotide. So what is the difference between nucleoside and nucleotide? Nucleoside is sugar plus nitrogen base, and nucleotide is sugar plus nitrogen base plus phosphate. So जनरली बच्चे है ना कंफ्यूज होते हैं एग्जाम के टाइम पे टाइड एंड साइड में. तो एक शॉर्ट ट्रिक दे रही हूँ, याद रखिएगा. न्यूक्लियोटाइड के स्पेलिंग में टी आता है, करेक्ट. न्यूक्लियोटाइड के स्पेलिंग में टी आता है, टी, राइट. एंड व्हेन ऑल थ्री थिंग्स आर टुगेदर, 3T, इट्स अ टाइड. सो 1 2 3 तीन चीजें साथ में है तो टाइड है, नहीं तो साइड है. कौन सी तीन चीजें हैं बेटा? पेंटोज शुगर, nitrogen base and phosphate group. When three things are together, tide, otherwise side. ठीक है, ये शॉर्ट ट्रिक बता रही हूँ, इजी हो जाएगा पेपर देखने के बाद, नहीं तो कंफ्यूज होते हो. यस, न्यूक्लियोटाइड, यस कृष्णा, यू आर राइट, हैज पेंटोज शुगर, नाइट्रोजन बेस एंड फॉस्फेट. परफेक्ट. If a phosphate group is also found esterified to the sugar, it's going to be called as nucleotide. Adenosine, guanosine, thymidine, uridine, cytidine are nucleosides. मतलब अभी तक phosphate attach नहीं हुआ है. phosphate attach होने के बाद हम उसका नेमिंग सेरेमनी चेंज कर देते हैं. अभी उसको क्या बुलाएंगे? एडेनिलिक एसिड, थाइमिडिलिक एसिड, गुआनिलिक एसिड, यूरिडिलिक एसिड, साइटिडिलिक एसिड्स. ओके. जैसे कि आज अगर मैं तुम्हें बुलाऊं, तुम्हारे नाम से बोल रही हूँ, कौन है अभी स्क्रीन पे मेरे, कौन किसके नाम है? भूमि, कृष्णा, तो मैं तुम्हें नाम से बुला रही हूँ, करेक्ट. अब अगर जाके तुम लोग आगे जाके डॉक्टर्स बन गए तो तुम्हारा नाम के पहले एक प्रीफिक्स लग जाएगा। तो क्या बुलाना पड़ेगा? डॉक्टर भूमि, डॉक्टर कृष्णा, हाँ, और कौन है मेरी स्क्रीन पे अभी इधर? डॉक्टर वैष्णवी। तो ये जैसे नेम चेंज मतलब ऐड हो रहा है कुछ, तो इसीलिए तुम्हारा ओवरऑल क्या कहते हैं, एक डेजिग्नेशन चेंज हो रहा है ना, वैसे है। सो न्यूक्लियोसाइड्स आर कॉल्ड एज एडिनोसाइन, गुआनोसाइन, है ना, ये देखो, थाइमिडीन, यूरिडीन, साइटिडीन। और अगर फॉस्फेट ग्रुप अटैच हुआ तो उनको और एक डिग्री उनकी बढ़ गई, देन एडेनिलिक एसिड, थाइमिडिलिक एसिड, गुआनिलिक एसिड, यूरिडिलिक एसिड, साइटिडिलिक एसिड, ऐसे बोला जाता है बेटा, ऐसे बोला जाता है। सो प्लीज डोंट गेट कंफ्यूज्ड विद द नेमिंग सेरेमनी। न्यूक्लियोसाइड्स के नाम क्या है और टाइड्स के नाम क्या है? तो अब जब हम कहते हैं एडिनोसिन ट्राइफॉस्फेट, करेक्ट, सो एडिनोसिन इज अ न्यूक्लियोसाइड एंड विल हैव 3 फॉस्फेट, इसलिए हमने उसको क्या बोला? ट्राइफॉस्फेट। ओके, सो यू कैन ऑल्सो कॉल इट एज adenosine monophosphate, or you can call it as adenilic acid. You can call it as thymidine monophosphate, or you can call it as thymidilic acid. Synonyms है वो, ठीक है? तो नेमिंग सेरेमनी में कोई गड़बड़ नहीं करेगा बेटा, हाँ, वेरी इंपॉर्टेंट। Then why can't we call it as if it has phosphate? It is a nucleotide only बच्चा, but each, जैसे कि adenosine is a nucleotide, even guanosine is a nucleotide, then it's a nucleotide only, but how will it stand out that now it is a nucleotide? What will make you understand that it is a nucleotide? All are nucleotides now, all are going to be added with phosphate group. So all are nucleotides. So nucleotide के example क्या हो जाएंगे? Adenilic acid, example of nucleotide. Adenosine, example of nucleoside. So adenilic, guanilic, thymidilic, cytidilic, it's specifically for that particular nitrogen base. इसीलिए all are nucleotides only. Obviously एक बार phosphate group add हो गया ना, तो सबको हम nucleotide ही कहेंगे, no doubt about it. But what would be the examples? Example of nucleotide cannot be adenosine. Got it? They are already like that way. They are already like that way only. Okay? So it is your phosphoric acid which will be added. No, beta. Phosphate group will be added from which source? H3PO4. ठीक है, इसीलिए वो एडेनिलिक एसिड, थाइमिडिलिक एसिड ऐसे कहा जाएगा। गॉट इट? अंडरस्टूड? एनी डिफिकल्टी? So nucleic acids like DNA and RNA consist of nucleotides only. DNA and RNA act as genetic material. They act as genetic material. Any difficulty in understanding this? What's your doubt? Hope you all have understood this, huh? Why do we use adenosine as a term? Why do we use adenosine term in ADP, ATP? It's going to be adenosine with one phosphate, so adenosine monophosphate. Two phosphates, adenosine diphosphate. 3 ATPs, adenosine triphosphate. I had actually explained these things in detail at Parle and Villeparle and Borivali. So इसीलिए like this is a thing that needs to be understood very nicely, that what do you mean by mono, di, tri, how much energy bonds are present in each of them, how are they going to be coming together, ओके, सो एडिनोसाइन का मतलब है पेंटोज शुगर है और उसके साथ अपना नाइट्रोजन बेस है। अब उसके साथ एक ही फॉस्फेट अटैच्ड है 5 प्राइम एंड पे। मैंने प्राइम क्या होता है वो भी एक्सप्लेन किया था इनफैक्ट, तो मैं आगे करूंगी ऑफकोर्स। तो फिफ्थ पोजीशन कार्बन पे इफ देयर इज गोइंग टू बी वन फॉस्फेट वी कॉल इट एज मोनोफॉस्फेट। 2 are attached, diphosphate. 3 are attached, triphosphate. So adenosine का मतलब होता है नाइट्रोजन बेस प्लस पेंटोज शुगर इज ऑलवेज देयर, नंबर ऑफ फॉस्फेट ग्रुप्स अटैच्ड वुड बी डिफरेंट। ओके, ADP or ATP will be a nucleotide because it's going to be what? Adenosine. D stands for what? Diphosphate. Even if it is AMP, adenosine monophosphate. So adenosine means what? Pentose sugar and nitrogen base. Okay, but adenosine के बाद तुम MP लगा रहे हो या DP लगा रहे हो या फिर TP लगा रहे हो, मतलब you are adding a phosphate moiety to it. So it is going to be a nucleotide. Got it? Understood? ठीक है? Why can't we call it as adenyllic acid? Adenyllic acid बोलूँगी तो will you come to know if it is having a single phosphate or 2 phosphates or 3 phosphate? Would you come to know? If I say adenyllic acid is present, so does it give you a clear idea about how many phosphates would be present? Though it's a though it's a nucleotide, agreed, it's a nucleotide. But how many phosphates are going to be present? Does it give a clear picture about this? Just using adenyllic acid word? No, na? Exactly. So to make the things very precise, we use the word monophosphate, diphosphate, triphosphates. But adenyllic acid, guanylate acid, cytidyllic acid, these are collective terms which are going to be specifically used in cytoplasmic content. So what are the cytoplasmic or nucleoplasmic content, to be very precise? Okay, so these are the terminologies that you need to understand. Yeah, someone has asked me explain the first point. I'll show you the figure, I'll show you the diagram, what do you mean by heterocyclic rings मैं बताऊंगी. Okay, तो और भी simple हो जाएगा ये. Adenine and guanine are substituted purines while the rest are substituted pyrimidines. So we have got 2 purines and 3 pyrimidines. Out of the 3 pyrimidines, you know that uracil is present in RNA, not present in DNA, whereas thymine is present in DNA, not present in RNA. Okay, इसलिए वो 3 हो जाते हैं. The skeletal heterocyclic ring is called as purine and pyrimidine respectively. So we have got a purine का ring and a pyrimidine का ring. आगे diagrams है तो easy हो जाएगा. The sugar found in polynucleotides is either a ribose sugar or 2' deoxyribose sugar. So ये जो ऊपर एक ऐसा कॉमा दिख रहा है ना, it is called as prime या dash, 2' या 2'. So what exactly is this 2', 2', 3', 5', 5', 3'? ये क्या होता है? So carbon of a sugar in a ring form will always be denoted as prime या dash. तो सेकंड कार्बन या सेकंड पोजीशन पे जो कार्बन है उधर एक ऑक्सीजन कम है, इसीलिए उसको डीऑक्सी कहा गया है. डीऑक्सी मीन्स 1 oxygen less. Okay, a nucleic acid containing deoxyribose is called as deoxyribonucleic acid, while the one which is having a ribose sugar will be called as ribonucleic acid or RNA. In a nucleic acid, a phosphate moiety is linked at the 3rd carbon of one sugar and 5th carbon of another sugar in a succeeding nucleotide sequence. So ये अगर अपना शुगर है तो इसके 3rd कार्बन को जो फॉस्फेट अटैच होगा, the same phosphate will be attached at the 5th position of the next sugar. Okay, so it is like this way, 3rd carbon and 5th carbon. इन दोनों के बीच में फॉस्फेट अटैच होता है. अगेन इसके थर्ड कार्बन और उसके नीचे वाले फिफ्थ कार्बन, उसके नीचे फॉस्फेट अटैच होता है. दिस इज द वे द न्यूक्लियोटाइड्स आर गोइंग टू बी अरेंज्ड. फाइन. The bond between the phosphate and hydroxyl group of sugar is called as an ester bond, peptide bond, glycosidic bond. वैसे ये क्या है? Ester bond. Fine. As there is one such ester bond on either side, it is going to be called as phosphodiester. I'll explain. Don't worry. I know ये थोड़ा visualize करने के लिए आपको difficult होने के chances है, but don't worry, I'll explain. Okay. So I'll just come to this first. See, here you can see this is going to be your adenine, which is a heterocyclic ring. So someone was asking me about what is this heterocyclic ring. So see, this is your adenine, which is a heterocyclic ring. This is your guanine, which is also a heterocyclic ring. These both are purines. Okay, and look over here, this is a double ring structure. Purines are double ring structures. You can see from the diagram itself. Whereas look at the pyrimidines, beta. They are single ring structures. Cytosine, uracil and thymine. These are single ring structures. So pyrimidines are monocyclic while purines are dicyclic. ठीक है? So pyrimidine bases are single ring monocyclic nitrogenous bases which are made up of cytosine, thymine and uracil. These all are pyrimidines. Purines are double ring structure, nitrogenous bases, adenine and guanine. So now you all understood what is the meaning of this. समझा? Okay. Now there was a person called as Chargaff. So Erwin Chargaff has given some important breakthroughs in this ATGC pairing. So Erwin Chargaff in year 1950 estimated the relative amounts of the four nitrogen bases, that is adenine, thymine, cytosine and guanine in DNA. I told you DNA में uracil नहीं होता है. तो DNA में क्या होता है? A, T, C, G. They don't have U. They observe that pyrimidine and purine always occur in equal amounts of DNA. तो DNA में जितने A हैं उतने T रहेंगे और जितने G है उतने C रहेंगे. It's a thumb rule. Okay. They also found that the base ratio of A plus T upon G plus C may vary in DNA of different group of animals and plants, but A plus T upon G plus C ratio remains constant for a particular species. So when you talk about these values now, so you get specific ratios. So that ratio will remain constant for a species. So if you get a DNA sample of unknown source, you don't know ये किसका DNA है, तो जब तुम A plus T by G plus C निकालते हो, उसका रेशियो निकालते हो, तो वो जो वैल्यू आएगी वो वैल्यू को देखते तुम्हें समझ में आएगा ये जो डीएनए है ह्यूमन का है, ये डीएनए रैट का है, ये डीएनए मस्कीटो का है, ये डीएनए कॉकरोच का है, XYZ. So every species has got its own specific value. Okay, got it? Fine. So this is your Erwin Chargaff's rule. Before this, fusion showed that the chromosomes contain DNA. He established DNA. He established that DNA contains two pyrimidines, cytosine and thymine, and two purines, adenine and guanine. Wilkins and co-workers showed that purine and pyrimidine bases are placed regularly on DNA molecules at a distance of 3.4 Armstrong. ये जो A है ना, A के ऊपर ऐसा एक डिग्री का सिंबल डाला है. It is read as what? Armstrong. ठीक है? DNA is composed of sugar molecule, pentose sugar of deoxyribose type, phosphoric acid, also called as phosphate when in chemical combination, nitrogen containing bases. These are nitrogen containing organic ring compounds. Did you all understand what is it exactly? Fine. So this is about in general nucleic acid. We have to study the structure of DNA and we have to study the structure of RNA also and types of RNAs also. But it's almost 6, so I won't extend the lecture beyond this. Okay, so in the next class I'll go ahead with structure of DNA. which will help you all to understand the things or the points which I have discussed just now, 3 prime and 5 prime end, जिसमें आपको confusion है कि exactly क्या होता है, वो भी आपको बहुत easily समझ में आ जाएगा when I'll explain the DNA structure. You will be clarified with 5 prime end, 3 prime end, phosphoester bond, phosphodiester bond. Everything will be crystal clear in your mind. So next class is very important. Kindly do not miss that class. I'll be going in detail about structure of DNA, which is almost, from this structure of DNA in your need, almost 3 to 4 questions are asked every year. So you can imagine 4 questions aate to 16 marks, 16 marks on the structure of DNA. So it's going to be a very important lecture in the next class. So kindly do not miss it, beta, and pay attention very carefully when I'll be explaining the structure of DNA. It's going to be repeated again in molecular biology. Okay, fine. Sure. Have a nice day. Have a nice evening. Bye bye. See you all.